Welcome everyone to our physiology class. I believe everyone has seen the plot of blood recognition in TV dramas. From a modern perspective, you must find it quite absurd. But does it really have no credibility at all? Today, let's explore the secrets behind blood recognition together. Let's recall the plot. In the process, the criterion for judgment was whether the blood could mix. As shown in the diagram, the first scenario shows no mixing. The second scenario shows mixing. So why does this difference occur? This takes us back to the year 1900. At that time, the famous Austrian physician Karl Landsteiner was working at the Vienna Institute of Pathology. He observed that in blood transfusion therapy, sometimes transfusions between two people of different races, genders, and with no blood relation were safe. But other times, even transfusions between father and son, mother and daughter, or siblings, could cause the death of the recipient. To find the reason, he conducted an experiment that seems very simple by today's standards. Mixing one person's red blood cells with another person's serum and observing the result. The results were puzzling. The same person's red blood cells showed two different reactions in different people's serum. One was uniform distribution, presenting a consistent light red color. The other was agglutination into red clumps, which stood out distinctly against the pale yellow serum. So, Landsteiner compiled the experimental results from 22 people into a table, hoping to find a pattern. Through careful analysis, he proposed the prototype of the ABO blood group system. He divided the blood types in the table into three types, A, B, and O. Mixing blood of different types led to different outcomes. Agglutination or hemolysis could occur. If this happened inside the human body, it could be life-threatening. Later, by expanding the sample size, Landsteiner ultimately determined there were four blood types, A, B, O, and AB. In 1927, after international recognition at a conference, his original letter naming system was adopted. From then on, the modern blood group system was formally established. For this significant contribution, Landsteiner was awarded the Nobel Prize in Physiology or Medicine in 1930. Since his discovery of the first human blood group system, the ABO system, to date, we have discovered 35 different red blood cell group systems. Among them, the most clinically relevant are the ABO and RH blood group systems. Next, let's focus on the ABO blood group system. Based on the presence or absence of A and B antigens on the red blood cell membrane, we can classify blood into four types. Those with only A antigen are type A. Those with only B antigen are type B. Those with both A and B antigens are type AB. And those with neither antigen are type O. People of different blood types have different antibodies in their serum, but they do not contain antibodies corresponding to their own red blood cell antigens. Therefore, people with type A blood have only anti-B antibodies in their serum. People with type B blood have only anti-A antibodies. People with type AB blood have neither anti-A nor anti-B antibodies. And people with type O blood have both anti-A and anti-B antibodies. When we studied cell membrane structure, we learned that in the ABO system, the antigens on the red blood cell surface are determined by different sugar chains. So what determines the sugar chains on the red blood cells? The answer is genes. Both A and B antigens are formed on the basis of the H antigen. Under the control of the A gene, cells produce the A enzyme. This enzyme attaches an N-acetylgalactosamine to the H substance, forming the A antigen. The B enzyme, synthesized under the control of the B gene, attaches a galactose to the H substance, forming the B antigen. Type O red blood cells lack the genes to produce A or B enzymes, so they lack A and B antigens, but still possess the H antigen. In fact, the H antigen itself is formed on another precursor substance containing four sugar groups. Under the action of the fucosyltransferase encoded by the H gene, fucose is attached to the precursor substance, forming the H antigen. If the H gene is defective, the H antigen cannot be produced, and consequently, A and B antigens cannot be formed. This blood type is known as the Bombay phenotype. Therefore, genes determine which glycosyltransferase is produced, which then catalyzes the attachment of specific sugar groups to specific positions on the precursor substance, ultimately controlling the composition of the oligosaccharide chain that determines blood group antigen specificity, and thus determining the blood type phenotype. The distribution of blood group antigens in populations varies by region and ethnicity. The inheritance of the human ABO blood group system is controlled by 3 alleles, A, B, and O, located on chromosome 9. On a pair of chromosomes, only 2 of these 3 genes can be present, 1 inherited from each parent. These 3 genes can combine to form 6 genotypes. Since A and B are dominant genes and O is recessive, there are only 4 blood type phenotypes. People with the same blood type phenotype do not necessarily have the same genotype. For example, a person with phenotype A could have genotype AA or AO. But a person with phenotype O can only have the genotype OO. Using the principles of blood type inheritance, we can predict the possible and impossible blood types of offspring. Therefore, parental relationships can also be inferred from the offspring's blood type phenotype. However, it is important to note, in forensics, when using blood types to determine paternity, only negative conclusions can be made, not positive ones. Now, let's return to the question posed at the beginning of the class. Perhaps blood recognition does have some scientific basis to a certain extent. It's just that the ancients, limited by the technical level of their time, did not discover the underlying genetic principles.